Okay, let's get our final didactic started. And it's a real pleasure to have Andriy here to present Embracing Complexity of the Evolution of Targeted Therapy Resistance. And resistance obviously is a topic we're all very interested in. And Andriy is an experimentalist but he's someone who thinks like a modeler, and he's someone that works closely with modelers. And there's only a few of those people in our community, and so we should cherish them. So thank you, Andriy.
Thank you, Sandy, for your very nice introduction. Okay. Is it on? Oh, yeah, it's on. So thank you, Sandy, for your generous introduction. And I thank Sandy and Christine for this opportunity to present our work. And I thank everyone who decided to stay till the last day. So I'll do what I can, trying to not to disappoint. And especially appreciate the opportunity to speak, to give us this didactic talk that allows to focus more on the conceptual big picture without getting bogged down into the technical details. So, as you all know, the molecular biology revolution has led to the introduction of so-called targeted therapies, so small molecule inhibitors that can suppress oncogenic signaling nodes. So these drugs are not available for all cancers, but for cancers with targetable oncogenic addictions, you can get spectacular responses as exemplified in this clinical case of subcutaneous melanoma patient treated with BRAF inhibitors. And as you can see, all these multiple metastatic lesions are essentially melting away. Cachexia is gone. So things work great for a time, but unfortunately the tumors go back in the exact same location and eventually the patient perishes. So this example really illustrates the problem that we have across the board of all types of targeted therapies in different contexts. So the magnitude of response can vary. The duration of the remission could vary from weeks to months to even years, but almost without exceptions, resistance comes and cancels the effect of the therapies, leading to the demise of the patients. So why is this happening? At the most basic level, there are two main causes. So first, our therapies fail to eliminate all of the tumor cells. So some of the subpopulations of tumor cells persist within the residual disease. And then, if that was the only problem, it would be okay. But the problem is that these persistent residual populations evolve and develop resistance, which lead to the eventual relapse. So how do we deal with the problem? Naturally, given the initial success of targeted therapies, the main effort is within the molecular biology, molecular oncology field, is to identify the molecular mechanisms that allow the tumor cells to survive under therapy and identify resistance mechanism and hopefully develop new drugs that could cancel them out, leading to eradication and reverting sensitivity to therapies. So this effort has resulted in a large and growing list of targets, led to the development of new drugs, and this gave some substantial improvements, but mostly by improved targeting of some of the on-target mutational mechanism, extending the duration of responses, but not really resolving the problem of inevitability of resistance. And as we know here in the community, another big effort, well, which pales, of course, in the scale compared to the mainstream, but it still has gathered some support, and the support is growing, is the idea of to use the existing drugs, but apply them in a different way that considers the evolutionary responses within tumors and tries to eliminate the residual disease through multiple strike therapies or trying to extend the duration of responses. And there are some good causes for optimism, but I would say the jury is still on, and it's not really as slam dunk as we might have hoped initially. So there are multiple challenges. So how do we solve the problem of resistance and the problem of acquired therapy resistance? And I think like in order to solve the problem, we have to start with correct conceptual maps. So if you are to read any mainstream review in the area, you would be left with the impression that we have figured out all the main things that we need to know, and what is left is to figure out these details, identify those targets, develop the drugs, or perhaps improve your models to be more accurate and be more capable of guiding therapies in clinic. But I would say that our current understanding is more like this medieval world maps that actually capture some of the features with reasonable accuracy. Other features may be a little bit out of proportion or a lot out of proportion, and big things like Americas or Antarcticas are entirely missing. So to get to the right results, I think we need to improve our conceptual maps. And of course, like map is not a terrain, any map is an abstraction, but I think there is a big room for improvement to guide our efforts. So what do we know about how resistance develops? So essentially, there are two major ways of thinking about the potential cause of evolution of resistance. The first model that we've heard a lot about from some of the presentation is so-called competitive release. So the idea that before you start the treatment, you have a subpopulation of resistant cells already present in the tumors. So when you start the treatment, you eliminate the sensitive cells, resistant cells are expanding due to competitive release, which leads to relapse. And then if the resistance is linked with baseline fitness penalty, you might be able to explore the evolutionary dynamics with adaptive therapy. Another model that has a lot of empirical support is that you don't necessarily have pre-existence of full resistance. Instead, a subpopulation of tumor cells might be able to so-called like persist, basically survive, and having some limited proliferation, but barely hanging there, providing the pool for the residual disease. And then eventually some of these cells will acquire true resistance, leading to relapse. So how do they transition from sensitivity or persistence to resistance? And again, there's not much in terms of the kind of explicit elaboration on this topic, but if you read between the lines, the implicit assumption that's fairly common is that you get from persistence or sensitivity to resistance through a single either mutational step or epimutational switch, essentially epigenetically plasticity-mediated state transition that gives you resistance. And then basically, it's essentially a saltationary kind of quantum leap. And I apologize for the low resolution. Somehow it got messed up with the transfer of the slides. So why aren't we there yet? Why aren't we facing these challenges? And what are the challenges that we are facing? So the main kind of reasonably embraced and recognized reason is the problem of both intra and intertumor heterogeneity. So since cancers are a result of the somatic evolution, which has a substantial stochastic component, essentially every tumor, every cancer is unique evolutionary snowflake. And so the resistance mechanism, persistence mechanisms that are found in one tumor are not necessarily generalizable to the next tumor. Moreover, within each tumor, you have typically quite a degree of phenotypic and genetic heterogeneity. So even if you figured out how to kill off a major subpopulation of tumor cells, you might still have to deal with other subpopulations that are less sensitive to the treatment. But still, the idea that by improving our diagnostic tools, we might be able to whack these guys one by one and eventually lead to the cures without changing too much in terms of our approaches. However, there's another part that's, I would say, gets some recognition, but not as much recognition as it deserves, is that the evolution is not limited to expansion of pre-existing variants, because otherwise the evolution of diverse life on Earth didn't come from the selection of the pre-existing forms within the primordial billion years ago. So the residual populations can create new evolutionary innovation, can create new types, which presents a big challenge in terms of this approach of identifying and eliminating clonal populations and to deal with the resistance. So what I'm going to share today is kind of results of our lab's effort that identified new, I think, important dimensions to the problem of resistance that I think we need to consider in order to improve our odds of achieving success. And I was really drawn to Moffitt by fascination with the idea of evolutionary informed therapies, adaptive therapies, and other types of therapies. But I also kind of thought that there might be a missed opportunity of really kind of having the intermediate stage of integration of experimental and mathematical modeling before going to the clinic. And Maxi and Luciana provided a very nice plug for the utility of this integration in the first day. So the point is that instead of hoping that our assumptions, brilliant assumptions, are correct and running right to the clinic, the use of preclinical models integrated with math models really allows us to stress test our assumption, make sure that we truly understand what's going on, and then we refine our both conceptual understanding and modeling tools until we really can predict how tumors respond to therapies in mice. And of course, mice are not tumor. You would need recalibration, but it would provide really a basis for much more solid understanding because you have a great degree of control. You can't play this Groundhog Day scenario where even if you are wrong, you can go back and revise your assumption and retry until you achieve satisfactory outcomes. And we decided to focus on ALK-positive lung cancers initially because they're really good poster children of the both success and limitations of targeted therapies. So there are good drugs that can induce very long remissions in the clinic. There is a wealth of understanding of the underlying molecular mechanism, so we don't have to do much of the molecular discovery. And we also have fairly good experimental tools, both in vitro and in vivo models that recapitulate both the initial response and then eventual relapse. So one thing that we found, again, and this is, I think, the case for the vast majority of experimental models that do show decent initial sensitivity, is that you don't have a pre-existence of full resistance. So if you hit the truly resistant cells with the drug, they don't care. They would form within 1 or 2 weeks, these huge colonies. Whereas if you hit the naive cells, you get plenty of these tiny colonies, some of which over time will start growing at higher rates. But so essentially you start with persistence. And then the question is, okay, how do these transition from persistence to resistance? Again, the assumption is that one hit, either mutational or a mutational switch, should be enough. So if that's the case, we reason that we should be able to capture it in a very simple experiment. So we are growing cells in the presence of ALK inhibitors for various duration of time, and then harvest, get them to single cell suspension, and seed for the clonogenic assays. So if transition has occurred from persistence to resistance at any time during this culture, you would expect to get a large colony. And so with longer time, you would expect to see a kind of higher proportion of large colonies, but you would expect to see sort of a bimodal distribution. But what we found instead was different. So we had a predicted increase in the proportion of clonogenic cells, but then the size of the colony, there was a big variability, but you might appreciate there was basically a slow creep in the sort of movement of the average size and movement of the distribution that suggested that we might be dealing with a more gradual adaptation rather than this sort of quantum switch. However, the caveat is that it's possible that the transition would happen during this clonogenic phase and then it would blur the picture. So really to get to the bottom of it, we decided to use essentially a digital twin approach and have a kind of in silico avatar of our experimental assay where we basically fix the initial and final distribution in the resistant cells. And what basically we task model to do is to transition to resistance either through a single step, as people assume, or through various number of intermediate steps. And then we can compare these intermediate steps between what is predicted in the model based on the number of steps to resistance and our experimental observations. So, and what we've found is could be recapitulated in this example where basically no matter how we try, the single step transition didn't really match our kind of experimental observations. And then if we allow resistance to develop through multiple steps, we get a fairly good fit, and we basically see it across a wide range of biologically relevant parameter space. So the implication is that the resistance appears to be developed through a kind of gradual process instead of quanti leap. And so, which means that resistance is not a switch, it's a path. And it's a path, again, like I don't have time to go into details, but we have looked carefully in the underlying molecular mechanism. And so we see that essentially a path through both adaptive and epigenetic landscape. So we're talking about plasticity, and we're seeing a huge contribution of the epigenetic changes, but we also see that the very same cells acquire some copy number changes that are contributing to resistance. So it appears that the resistance path is more complex. It involves both plasticity mediated and genetic changes that add up and synergize with each other. And we've tried to make sense of it in this review in Trends of Cancer, but recent evidence from Arjun Raj lab and from Itai and I lab suggest even more interesting and complex picture. So importantly, our findings were done essentially in single experimental system, but there's recent study from Itai and I group that looked at PARP inhibitor resistance, but also looked across a wider range of targeted therapies, where they essentially seeing an identical picture which we have observed. So I believe what we have uncovered there is generalizable across the wide board of resistance context. And in fact, our model doesn't exclude that to have one single major resistance mechanism, it could be viewed as a particular case of the sort of more of a gradual scenario, but not the other way around. So why should we care about it? So from the standpoint of a kind of more traditional approach, what it means, if your resistance is made up of multiple building blocks, the fact you identified one and figure out how to target it would mean that you could improve responses and you could revert, get back some of the sensitivity, but it would mean that you don't necessarily fully destroy the foundation of resistance. And this is consistent with what we see, like in any given experimental system, there would be dozens of papers that describe different resistance mechanisms, and all of them can show statistically significant improvement of responses, but it seems to tell that there might be present at the same time in multiple subpopulations or even within the same cells. And from the standpoint of our evolutionary informed therapies, the point is that if we use mechanistic models to predict responses, but these mechanistic models start with wrong assumptions, either assuming pre-existence of resistance or single-step transition to resistance, you might be the most brilliant mathematician, but if you start with wrong inputs, your model cannot transform wrong inputs into correct predictions. So it's important to approach cancer with epistemic humility and overly excited about the brilliant idea, hoping that the reality will conform to it. But so far, what I've shown was mostly done in vitro, and as we know, and there are a number of cool talks, like from Mehdi, that pointed out the importance of microenvironmental considerations. So different ecological habitats within the tumors exist within gradients of nutrients, oxygens, etc. And this all create heterogeneity of context that can potentially influence sensitivity to therapies. And one most obvious thing is, and our own Bob was one of the first to really bring it to the major discourse, is the potential for the microenvironmental induced resistance. So it's known that multiple components produced by non-tumor cells within tumor microenvironment, like elements of extracellular matrix and multiple cytokines, can dramatically blunt sensitivity of tumor cells to targeted therapies across essentially all context of targeted therapies. So, and it leads to kind of a little bit of a split scenario. So on one hand, we know that we can take essentially any well-known experimental model, we'll use experimental model, subject them to therapies, we eliminate the majority of cells, but a subset of cell will persist, and these persisters, if you give them enough time, they will develop resistance. So the cells are capable of developing of persistence and resistance through entirely cell-intrinsic mechanisms. At the same time, if you co-culture these very same cells with stromal fibroblasts, typically what you get is that you have a much larger fraction of the cells that can survive, and then these cells are capable of proliferating at much higher rates. So this very same cells that can survive and persist and resist on their own, they can get a benefit from this help from the non-tumor components of the tumor microenvironment. So it leads to the obvious question, okay, what's going on in vivo? And if you read the literature, typically people ascribe whatever they see to a single drug, to a single mechanism, either from the cell intrinsic side or from the environmental side. But a priori, it's sort of hard to reconcile why would you only have one but not the other? So we've tried to understand the relative contribution of cell intrinsic and environmental effects in the responses of tumors in xenograft models. And again, I don't have time to go through too much detail, so I'm going to share the, I think, most informative insight that of the patterns of cell proliferation within the tumor. So these targeted therapies have very strong cytostatic components. So before the treatment, you have plenty of proliferation. Here you're looking at the BRDU that captures the cells in the S phase of the cell cycle progression. And you start treatment, you start seeing the emergence of the remarkable spatial patterns. As the longer you treat, the lower the proportion of proliferating cells becomes. And then all of them are essentially you find them almost exclusively in the proximity to the stromal regions. So this is where your fibroblasts are that are producing these extracellular matrix and growth factors, et cetera. And which basically indicates that the stromal effects could be the major cause of in vivo persistence. But surprisingly, even at relapse, so at relapse, basically, now you find proliferation outside of the peristromal niche as well, which indicates that the relapse is mediated, at least in big part, by acquisition of cell intrinsic resistance. But even upon relapse, we still see this residual bias that indicates that relapse and resistance is a combination of both microenvironmental and cell intrinsic effects. So Tatiana and under David's supervision has developed a digital pathology pipeline with involvement of Daria from our lab that enables us to kind of robustly quantify these effects using metrics taken from the field of spatial statistics, which can really provide more of a robust quantification, and we can then use these parameters for the parameterization of the models. So which basically we can capture both the distance of this stromal effects and the magnitude. And again, sort of it confirms the visual picture that we can get just from looking at the tumors that indicates that this stromal effects appear to be the major cause of the in vivo persistence. So again, like I've shown you on a single model, but and it's sort of it could be tricky if because this approach requires some like big inequality in terms of how close tumor cells are to the stroma, but to the extent that we can tell, we see some evidence of this effect throughout any experimental system and in many clinical samples that we've analyzed. So again, the point is that even though the subset of tumor cells are capable of survival persistence through cell intrinsic mechanism, in vivo, the majority of the survival comes from tumor cells being close to these peristromal niches, which buys them time to essentially provides them with an ecological rescue mechanism that buys them time and facilitates the transition to the evolutionary rescue. So one important part is much like with the cell intrinsic resistance, we see that this microenvironmental effects cannot be reduced to a single mechanism. So instead, they reflect a simultaneous action of multiple juxtacrine and paracrine interactions. So one way to conceptualize it, you can think about your oncogenic signaling as this kind of bright bulb that illuminates the cells. And if you're applying the targeted therapies, you turn off this bulb. But now if you have a cell positioned close to stroma, at the absence of treatment, the resistant fraction remains the same or even increases slightly. But treatment really induces competitive release, leading the resistant cells to dominate the tumors. And it leads to almost kind of instantaneous relapse. Within 1 to 3 weeks, the tumors start growing back. However, it's still possible that in realistic scenarios, in real tumors, you might have a much lower proportion of resistant cells. And so we wanted to use the modeling to really capture the competitive dynamics of sensitive resistant cells to see what would be the consequence of having a single resistant cells as we start the treatment. Could it still be compatible with very long remissions that we observed clinically? And what we basically found is that in mixed tumors, if we parameterize the model based on the inferences and proliferation and death dynamics from tumors of single type, we see a kind of big mismatch between the predictions and observations. So even the resistance fraction increases dramatically, we still would predict it even to be even higher, which suggests that there must be some sort of positive interaction where the resistant cells somehow help their sensitive counterparts. And so if we introduce in the model the interaction term, assuming the direct protective effect, we can fix the discrepancy, but also predicts very specific spatial patterns that we should be able to see in the tumor. And of course, being able to fix things in the model doesn't mean that we've really uncovered what's going on. So we looked at the patterns, spatial patterns in the tumor, and what we see here is quite closely resembles the previous picture that I've shown with patterns of cell proliferation. So essentially, the resistant cells outcompete sensitive cells outside of the stromal niches, but close to stromal niches, it's about 50-50. So being close to stroma increases fitness of otherwise sensitive cells, enables them to compete with the resistant cells, and it leads to these spatial patterns that we can capture these tools that I've shown you before. But it doesn't explain the interaction by itself. So what we think what's going on is that as you start the treatment, so the fraction of tumor cells close to the peristromal niches are protected. But as you debulk the tumor shrinks, and so you start losing these protective niches. However, if you have pre-existing resistance, their expansion limits the loss of the niches, and then it adds new niches, essentially kind of boosting this ecological rescue mechanism. So we have this indirect stroma-mediated interaction that I haven't seen any example or prior evidence in the literature, but I think it could be relevant not only in this system, but in some other contexts as well. So it looks like I'm running out of time, so I'll skip through some of the parts. But basically, if we incorporate these effects into the models, we can now capture both temporal and spatial dynamics that we observe in the experiments. And more importantly, now we can make some non-intuitive predictions and really stress test the model by challenging to predict how things would behave under different initial proportion. And we can get some kind of non-intuitive prediction of the kind of this crossing that we can then validate experimentally. So this model is not necessarily qualitatively accurate, but I think it captures the gist of the dynamic that would be completely missed if you. So a couple of additional points that I don't have time to elaborate, but I think they add to the new dimensions to the complexity of resistance. One thing, and I think it was touched by in several previous talks, is that as the tumors develop resistance, they then tend to be not necessarily more resistant to orthogonal stressors, but we're getting the tumor cells that are more capable of adapting faster to new stressors. And I think part of the reason is that the selection for resistance leads to indirect selection for increased plasticity, and then basically transitioning to resistance through multiple steps essentially amplifies, essentially working as a compounding effect that amplifies the selection, indirect selection for increased plasticity. And then work of Gosha here demonstrated that basically the being in the survival bottlenecks also gives stronger selective advantage to higher mutation rates. So we might also have an indirect, most likely stress-induced mutagenesis. Another important part that's, I think could be a really huge elephant in the room that's missed currently is that the impact of variability of pharmacokinetics. So as we get the drugs in the body, they're breaking down by the liver enzymes. And this is a very important mandatory part of drug analysis before the introduction in the clinic. And then once the drug are in clinic, oncologists tend to ignore the variability. Yet between individual patients, you can get orders of magnitude of differences in the activity of liver enzyme. So from our kind of initial analysis, it appears that at least a reasonable subset of cases of clinical resistance could be caused by the activation of the drug metabolism. So not tumor intrinsic, but caused by the differences in the drug exposure. So with that, it looks like I'm out of time, and Sandy is giving subtle visual cues. So just a few take-home messages. Yeah, I think there's still kind of a good deal of work we need to do to make sure that our conceptual maps are correct. And all these different aspects that we're talking about, plasticity, genetic mutations, they're all kind of really part of the same reality. And it's important to have an open mind and epistemic humility to recognize the complexity and beauty of the reality and trying to kind of really understand what's going on before jumping the gun. So yeah, resistance is a path, not as a switch. It has multiple components, and we need to consider them, especially if when we're building our beautiful models of trying to predict how things work. And with that, I really like to thank people in the lab and our many collaborators, both from IMO and outside, and Sandy and David and Noemi and all the people in the lab. Thank you.
Well, thanks, Andriy, for making resistance even more complicated. If it isn't complicated enough, but that's the reality of it, right? Do we have any questions? There's Mehdi.
Beautiful talks, Andriy. Sandy. I slept 2 hours last night. So Andriy, like I love this ecological rescue idea, which is amazing. But I mean, people try to deplete the fibroblasts 100%. You know, in under drug or no drug. This is the David Tiverson lab work on all this thing. and the tumors just still even grow faster and
Yeah.
evolve, right? So have you guys tried doing that? Or like, what do you think, if you haven't tried, what would you think would happen if you take out the fibroblasts totally?
So basically, by depleting fibroblasts, you're removing the spatial constraints essentially. And in those experiments, the tumors kind of in like Rego Kaluri lab experiment, they basically kind of go everywhere and start metastasizing. So I think it's kind of it's wrong to consider fibroblasts of their friends or foes. They're doing multiple things. Some of them kind of constrain tumors and important from kind of
talk. I have a question. What do you think is the relationship between these persister states that you talk about and other states that we also talk about, such as senescence or quiescence or stem-like cells, which people are identifying? Are these different costumes? Are they different routes along this?
Plasticity is essentially an innate property that allows cells to kind of rewire epigenetically, allowing them to kind of improve and acclimate to the drug and get better and better and better. So now, like quiescence is a different beast. It's especially important for like chemotherapies, where it's the cells could essentially wither the storm of the drug. It's not very helpful against targeted therapies because you have target present all of the time. So you go to sleep, you wake up, the drug is still there. It's not very helpful. If you go to the sleep, wake up, there is like a treatment break. Now you can expand and you can benefit from it. And senescence, well, the senescence classically it's sort of irreversible growth arrest. That's basically not a problem. The problem is that the senescence is associated with a secretory phenotype that could impact the microenvironment. Also in tumors, sometimes classical markers point to the senescence, but if you look at the cells, that then at some time they can transition to proliferation. So the senescence in cancers, at least with the traditional markers, could be a reversible process. So again, it's sort of all parts of the more complex reality. And even persistence, people like the term persistence, but it's sort of really a heterogeneous mechanism. And then I think sort of it's more of a continuum of resistance, from very weak resistance that we can label as persistence to more of a kind of intermediate and finally net positive cell growth we label resistance. But there could be, again, a huge range from like barely growing to a complete insensitivity to the drug.
Andriy, that was wonderful. Thoroughly enjoyed it. And I love the idea of this path instead of being a jump. But I guess my question is really more, I guess at this point, alternative hypotheses. The path could either be a single quantitative trait. So you just simply have, it's maybe quite simple. It may be a single trait that changes quantitatively, or it could be a vector valued trait in which it's several. So the analogy I'd say is it could be gazelles evolving to run faster in response to predators. That's just a gradual, nice quantitative trait. Or it could be gazelles growing horns to defend themselves, developing camouflage to not be seen and to run fast. So one is sort of a single trait, the other is multiple traits. What do you think?
Yeah, so I think we have kind of clear evidence of multiple traits. So it's the path is sort of involved. I mean, depending on how you define the trait, but the cool thing about path, where I think it makes things more complex. On one hand, we might sort of try to slow it down by targeting the phenotypic plasticity mechanism and mutational mechanism without having to think about specific traits. Another thing, sort of these intermediate stages can sometimes show unique collateral vulnerabilities that allow us to kind of really hit them and eliminate. So both kind of constrain their ability to change, but also kind of directly target this intermediate. I don't think it answers your question specifically, but I'll be happy to chat afterwards.